Dear students, welcome again. So, this is the first official lecture of the IIT Professor Assisted Learning in Biology. I am Aditya Mittal. I am a professor in the School of Biological Sciences at IIT Delhi, and I am part of a team of teachers who have introduced themselves to you in our previous modules. So, today this is the first official lecture towards building the foundations of what we think you require for learning biology the conceptual way. What you see in the slide, for example, here are various facets of biology. These are microbial cells, bacteria in particular. Next you see a plant and an insect, next you see a bird, and then next you see a human. Biology covers all these facets very clearly. We try to understand what makes them living systems, we try to understand what distinguishes them from each other, but most importantly we try to understand, is there a common theme between, let us say, this system, living system, and let us say this living system, which has come out of several cycles of what we know as evolution. We will talk about that in the coming part of the lecture. So, the first thing which we need to appreciate before we go into biology is that just like any science, any other science, biological sciences is a part of utilization of the scientific method. Now, the scientific method essentially means that let us say there is a student or a researcher sitting in Nagaland, and let us say there is a student and a researcher sitting in Arunachal Pradesh, compared to Ladakh, compared to Gujarat, compared to Orissa, compared to Kanyakumari. Now, these researchers are looking at some life phenomenon. Let us say they are looking at color of water in the raindrops. Now, if all of them observe that the raindrops are colorless, they will basically conclude that there is no coloring agent inside them. Now, will the conclusion be the same across our country about this observation? Will this conclusion depend on where the person or the researcher or the student is sitting? If it depends on those parameters, if everybody concludes a different amount of information from any observation, then that goes against science. The conclusion from the observation up to the conclusion, everybody stays what we call in normal jargon as on the same wavelength. That is the important part of scientific method. This is very important in terms of learning biology. Let me illustrate this in terms of the actual methodology that has to be followed. Let us say you have an observation. Now, to explain this observation, you would use a variety of explanations. Let us say I think something might happen like this, or you think something might happen like that, alright. These explanations about any observation, whether it was the apple falling from the tree which Newton observed and then coined gravity for you, or it is the color of seawater which gave our great C.V. Raman best insights into what is now known as Raman spectroscopy. These are aspects in terms of that we have observed something, and now we want to actually make sure that there is some kind of an explanation which is there, a reason why it is actually happening. This explanation we call as hypothesis. The plural of hypothesis, this I is replaced by E. So, let us have one hypothesis. Now, here I will go into an interesting historical aspect. Long time ago, science and philosophy were the same things. In fact, as you heard from one of our respected KV teachers, Aristotle is often called the father of biology. But remember, it was Aristotle who led to Copernicus's suffering, because Copernicus was opposing Aristotle in terms of an observation, a hypothesis for an observation based on the fact that Aristotle seemed to think that earth is the center of the universe, whereas Copernicus was absolutely convinced that earth revolves around the sun and earth is not the center, rather sun is the center. So, these two different explanations in terms of looking at the night sky, looking at the position of the sun, they were two hypotheses, and these hypotheses are supposed to explain how things happen. So, coming back to the fact, long time ago science and philosophy were the same things. In fact, one of the aspects is the highest degree in academics one can obtain is called a PhD, Doctor of Philosophy. That is because philosophy means thinking, and thinking means that you do not have to stick to one particular narrow minded attitude, you are basically open to all ideas. One aspect of the scientific method which is important for you to understand is, if there is any observation, be open to any explanation for it, then logically find the reasoning that may or may not explain it properly. So, after an observation we build a hypothesis. The aim of this hypothesis is to answer how. Classically, the aim of any scientific endeavor and philosophical endeavor is to answer why. However, we are trying to answer how, because we hope that after answering how we can answer why. Why is a much more difficult question, it is a deep question. So, the first thing is that we have an observation and we build a hypothesis to try to explain that observation. Now, once we have built that hypothesis, we need to do an experiment. Why do we need to do this experiment? Again, let us say there is a researcher sitting in Nagaland and there is a researcher sitting in Diu. These are so east and west of the country. Now, these two researchers are trying to explain, let us say, division of cells, how living cells are dividing. If they have an explanation, if they have a hypothesis to explain this thing, both of them should be able to do the same experiment in controlled settings and get the same results. Only then will both of them agree on why the cells were dividing in a particular manner. Let us say you have a horse reproducing and you have another horse which actually mates with a donkey, and you get a mule out of that reproductive cycle. Now, if somebody observes this, they will basically have a hypothesis that any time two different kinds of horse like animals mate, they will result in a different animal called a mule. However, the other side might think, they might have an explanation that no, it is not like that. You have horses mating to give horses only. So, even if one horse is involved, you will always get a horse. So, now observation of how reproduction happened or what kind of offspring was produced is built in this hypothesis. The experiment would be then that one would take horse to mate with another horse, and that would lead to production of horses only, a horse to mate with a donkey, and that will not lead to production of a horse ever, it will lead to a mule's production. So, this experiment must be done under controlled conditions. When I say controlled conditions, we can't leave wild horses and wild donkeys and just observe what offsprings come out of the jungle when they are in the wild. What we need to do is, we need to have a controlled environment where we know we have kept only one horse and one donkey. Of course, male female ratios have to be maintained. Based on the experiment, then we propose what is called a mechanism. Now, once the mechanism is proposed, the most important thing here is that this mechanism should be repeated and it should give rise to the same observation again. So, we started with an observation that this looks like a mule, it is neither a horse nor a donkey. Where did it come from? Well, it might have come from the fact that a horse mated with a donkey. Therefore, the mechanism of reproducing or getting a mule is for a horse to mate with a donkey, And so, if we find that this works, it is okay, but if it does not, let us say we assume that a mule came because a donkey mated with a donkey or a horse mated with a horse. In that case, we will have multiple cycles of horses mating with horses only and donkeys mating with donkeys only, and they will produce only donkeys and horses, they will not produce mules. In that case, our mechanism which we think that only horses are producing or only donkeys are producing, that will not work. Therefore, we go and build back our hypothesis. This cycle of creating hypotheses for observations, leading to experiments, leading to mechanisms is one of the foundations of any science and of course, biological sciences. Biology, life is considered to be a miracle because it appears to be a very challenging, complex system which has arisen out of we do not know what, probably a mix of chemicals. There are theories and theories to actually describe this. There are every religion actually has its own explanation of how life originated on earth. However, what is important within the scientific realms is to be able to understand the observations of life in terms of hypotheses, doing experiments to repeat to get at the mechanism by which some living systems are working or all are working, and if that mechanism does not replicate our observation, then we rebuild the hypothesis and go back in the same cycle until we actually come back to the same replication of the observation. This is the scientific method, it must be followed. Now, here I have to give a disclaimer. I am not trying to actually modify your thoughts or your beliefs in terms of what biology is, what life is and what you believe in. What I am trying to do is give you an objective way of thinking that once you have learnt what we are actually trying to accomplish here, once you have learnt how to build your own concepts, then you are free to decide, you are free to choose what explanation you have for a particular aspect of origin or for origin of life in general. This scientific method, my friends, is going to be the most important aspect towards your approaching biology as a conceptual science rather than a memorization science. Next, life as we know it on this planet is a mixture of large number of chemical reactions. Now, these large number of chemical reactions, they essentially originate from the basic principles of physics and chemistry, and the underlying theme or the law behind these is called the law of mass action. What is the law of mass action? Well, since class 7th, 8th, 9th, 10th, you have been told you have something called reactant which gives rise to product. So, you have reactants, let us say A and B reacting to give C and D. When A plus B react to give C plus D, and let us say this is a closed system, it is actually in equilibrium, then we write certain equations for this, right. So, the understanding of these equations conceptually is very, very important for you before you approach biology. Biology is a mixture of a lot of chemical reactions taking place simultaneously, and each of those chemical reactions obey this law of mass action. So, understanding this law of mass action in depth is very important for you. What is the law of mass action? While our esteemed colleagues in chemistry in the IIT PAL endeavor of chemistry will go into much more detail about chemical reactions, let me just give you a brief overview of what law of mass action is in terms of its importance for understanding biology as a scientific method. Let us say reactants A plus B react to give products C plus D. Let us say the forward rate reaction is K1 and the backward reaction is K minus 1. What law of mass action says is that in a closed system, the rate of concentration of a particular species of molecules, let us consider A in this case, In this case A is going to change how? Well, it is being consumed at the rate of k1, right. The tail of the arrow is facing A, therefore, it is being consumed. In that case, since it is being consumed, A is becoming less. It is becoming less at the rate of k1, which is multiplied by the concentration of A and B in the system. So, what we wrote in the law of mass action here is that the rate constant multiplied by whatever is on the tail of that rate constant, and the sign is on the tail of that rate constant. If A is getting consumed, the tail is there, therefore, the sign comes as negative, and the rate constant, the tail facing this rate constant is having A and B as the species. So, that is how we write it. On the other hand, now A is also coming from this back arrow. So, it is being produced, it is being added to the system with this rate constant. So, we write the sign of the rate constant is plus, we write k minus 1. At what this rate constant, the tail is facing C and D. So, it is actually, this is the rate of change of concentration of A in the system, and this is the law of mass action. Now, notice if we had equilibrium, capital constant K, which normally comes from here, that there is an equilibrium constant for a reaction. If we had equilibrium, in that case at equilibrium, rate of change of concentrations of molecules are the same, 0, because at equilibrium the number of molecules of A in that closed system, number of molecules of B, number of molecules of C, number of molecules of D, they all basically remain the same. There is no rate, there is no change in this thing. But remember, it is not that A is not reacting with B. If A reacts with B to give C and D, then C and D react to give A and B. So, the number of molecules are remaining the same. However, a particular molecule at any given particular time might have identity of A, B, C or D, but the number of A's will be the same, number of B's will be the same, number of C's would be the same, number of D's would be the same. So, this equilibrium, how to understand this? When dA by dt is equal to 0, then this equation becomes minus k1 A B plus k minus 1 C D equal to 0. Therefore, what you get is A B minus k 1 equal to minus k minus 1 C D. Therefore, you end up getting, what do you get from here? You get these signs go, is that right? And then you end up getting k 1 by k minus 1 is equal to this. Now becomes your association constant. K 1 is in the numerator, A and B are giving C D with respect to A B. So, this is your equilibrium constant, which is the ratio of rate constants between the systems. Now, this law of mass action is very important for you to understand. For example, how do I increase the production of C if I want to increase this production of D? If I want to increase the amount of D in the system, how do I do it? Well, I can increase the concentration of A or increase the concentration of B, then D will increase. How do I ensure that the reaction does not go back in the reverse direction? Well, I can keep doing this. So, this way I will have more production of this, it will still go back, but very small amount, this will keep going this, or I keep taking this D away from the system. So, now D is not a part of this closed system, Therefore, you have A plus B giving C plus D, and D is removed all the time. These are the classical tricks and techniques where the law of mass action is utilized in biological systems in your everyday life. As an example, let us take a simple example of glucose. Now, we go back to the PowerPoint slide here in a second to ask the question, what is the example for? All molecular decisions in biological systems are essentially a result of law of mass action. Now, this may come as a surprise to you. See, when you are deciding, for example, let us say you are hungry and you are walking on a road, and on that road you get a Y kind of a turn. So, you can go in the right direction or the left direction. You are hungry, which direction do you take? On the left side you do not smell anything, on the right side you smell some lovely pakoras are being made, you just smell fried food is being made or some pakora is being made which you like. Think about a food item. You just decide, okay, I am hungry, I can smell food, I will go in this direction. You made a decision, right. Now, this decision is happening because of some molecular reactions which are happening inside your system that allowed you to make this decision. Now, for you it is interesting and easy to think that I have a nose, I can hear, I can see, I can smell, and therefore, I am going in the direction of food. But believe it or not, a cell, a bacterial cell which you see here, which is 200 nanometer to 1000 nanometer, that is 0.2 to 1 microns big. This bacterial cell is also able to make decisions exactly like this, based on how much reactions are taking place in terms of the molecules which sense. So, let us come back to the drawing board and understand what that means. So, let us say all of you ate sugar, you must have eaten sugar in some way or the other, right. Bread, carbohydrates all of you know give you energy. Why? You have carbon, you have hydrogen, you have oxygen. Carbohydrate, carbon along with hydrate, water, right. So, these give you energy. This is generally what you know, bread, rice, roti. When you eat, the aim is to actually get carbohydrate energy. Where is this energy coming from? Well, it is coming from breaking of, let us say you have carbons, this energy comes from breaking of these carbon bonds. Now, think about this, that if I was to take an imaginary container of sugar and keep it over here, will that sugar in infinite time also break into carbon dioxide and water? No. On the other hand, you eat your bread over your sugar and you start exhaling carbon dioxide and water and you get energy. So, basically that sugar, out of which let us say glucose comes out, through a series of steps you come out and digest that glucose. In your breath, the breathing which you are having, you are coming out with carbon dioxide and water. As a side effect of this, you are getting energy. Now, this glucose is also utilized in your system to create a polymer called glycogen. Just like starch, glycogen is a polymer. So, in your liver cells, for example, glucose is stored as glycogen. Those of you who have been associated with any kind of athletics or sports must be aware that when you are running, then first you get very tired after some time, but then there is something called the second wind. If you just push yourself a little bit more, you start getting a second burst of energy, and then at the finishing time you try to get the third burst of energy, which is a very high energy system. This energy actually comes from a very interesting mechanism. First, the glucose in your bloodstream gives you energy. Cells chew up that glucose, get energy out, you are running. The first feeling of tiredness you get is when this glucose is finished. Now, once this glucose is finished, the glycogen, which is storage of energy in your cells, it starts breaking down. When glycogen starts breaking down into glucose again the same cycle of carbon dioxide, energy is done. Third cycle, when this glycogen also finishes, then you start burning your fat, and per unit mole fat gives a lot more energy compared to sugars. So, the important thing which I am trying to emphasize here is that when glucose goes into your body, there is a decision made that either it will go to CO2 plus H2O while giving you energy, or it will be stored as glycogen for emergency purposes. Now, when a glucose molecule is entering your bloodstream or is roaming around a cell or is inside a cell, the question you have to ask is, how is it decided what will happen to the glucose? How is it decided that glucose will go here or glucose will come here? Now, this is where law of mass action shows its beauty. What we wrote was, in terms of reactions, so let us say this A is actually glucose, and let us say for the energy pathway B with which glucose has to react in the first step is called BE, and for the energy pathway again for the storage pathway, now let us say S represents C and D here are CO2, H2O and energy, they represent these, and here you have let us say glycogen at some point of time. There are several steps which are taking, I am giving you a shortcut understanding of the concept here. Alright. So, how is this decision going to be made in terms of whether this glucose molecule comes here versus here? Think about it. Now, this is a concept nobody told the glucose molecule, nobody tells the cell you have to do this. So, think about it logically. Logically, if a single cell or my whole body for example is not having any energy, what will happen? I will become limp. If a cell stops functioning, then let us say I can't move my muscles. If I can't move my muscles, at some point my cardiac muscles which are moving to create a pumping effect for the blood to flow in my body will stop pumping, and in that case I will not get blood flow. If I do not get blood flow, I do not get nutrients and oxygen to cells all over the body, and therefore I die. So, the first importance is that my cells must have energy all the time. Therefore, glucose is going to always go into production of energy, and therefore I will be doing CO2, I will be producing CO2 H2O with breaking down of glucose all the time. However, interestingly, there is a point at which all the molecules of B, which is the first reaction here, which is going to take glucose here, all the molecules of B are let us say saturated with glucose. So, that means we have so much extra glucose in our system that whatever B can actually react with glucose is busy reacting with glucose. There is no more glucose which can come and bind to it. That means I have reached a threshold concentration of glucose which is saturating B. In that case I am still producing energy, but now I have excess glucose. In this case, this excess glucose, excess mind you, is going to go and react with S. Now, how is that possible? How does glucose know that I have to react with S only if I am excess? It does not. It simply is that the association constant of A plus B is high enough, is much higher compared to association constant of A and Bs. Therefore, what happens is that when there is a small amount of glucose, there is more affinity for that glucose to bind with B. This K1 is much higher than this K1 prime. Therefore, at low glucose concentrations, I am always going in the direction of production of energy along with carbon dioxide and water. The moment I have excess glucose, and the way you have to visualize it in your minds is that I am saturating all B. The moment I have excess glucose, now some of this Bs starts seeing. So, I need much higher glucose levels, and then I will, does not it look like normal household, that when we get, let us say we work hard, we get some money, we enjoy, we get good food, but if we have excess left, we store it for later usage. This is exactly how molecular decisions are made, based on a binding constant. So, you just today learned that the miracle of whether a glucose is going to go into energy production or it is going to be stored as a polymer, it is a miracle which does not involve any kind of complex understanding other than the law of mass action. A molecular decision is made based on reacting species which have different affinities towards reactants. So, in your subsequent lectures, when you are actually covering specific chapters of metabolism and catabolism, now if you think about metabolism and catabolism this way, rather than pressurizing yourself to think I have to remember this pathway or that pathway, this concept will make life a lot easier for you to appreciate, enjoy and understand metabolism and catabolism. Here as an example, catabolism, anabolism, right. So, you will basically get to see a variety of these things in terms of that. Now, we change gears a little bit. We have understood now, just to recap, we have understood that we must employ the scientific method. We have understood that law of mass action is something which we need to appreciate. This is the foundation of all reactions, combination of which is what we call as a living system. I have given you an example of how molecular decisions are made based on the law of mass action. Now, what is important for us to understand is the engineering, the key approach based on which this IIT PAL in biology is going to assist you. The engineering approach or the scientific approach towards understanding any phenomenon that we are interested in. You know, if I ask you how far are you sitting from the television if you are watching this broadcast, or how far are you sitting from the computer if you are watching this broadcast? Well, what will you say? Probably you will say 1 meter, 2 meters, 3 meters, maybe 5 meters, I do not know, but you will say that in terms of meters. You have been told that distance is measured in meters. You have also learned time is measured in seconds, minutes or hours. Well, I hereby ask you this question: how do you measure life? How do you measure life? What is, in other words, the question on the PowerPoint slide which you see, how do you define a unit of life? This is very important. As I told you, biology is a science. We must have a unit if we want to actually call some system a biological system. For example, you are assuming that the pen with which I am writing is a non-living object. Based on which? Based on which observation can you claim that this pen is a non-living object? The first thing which might come to your mind is it does not reproduce, because that is what people think about life in terms of. But tell me my friends, do not you know of life forms that are not able to reproduce? Mules, the example for which I gave earlier in terms of mating, mules cannot reproduce. We have a lot of parents who are looking for children, but they are not able to reproduce. Aren't they living? They are all living, of course. So, just saying that this cannot reproduce is not good enough to say that this is non living. Let us say you will say it cannot move, well it moved, and there are several species or several types of life forms which will be stationary, which during your period of observation will not move, but you do not say that they do not move. Basically what I am trying to actually drive the point at is that take your time to think about it, you do not have to actually answer this to us or to yourselves immediately. Take your time to think about and write down, jot down parameters that you think are important for life. The reason I am asking you to do this is then you must combine these parameters and define what a unit of life is. You will learn this in detail as the respective lesson on cell is covered to you. In general, we call a cell a unit of life. Now, basically the concept is that cell is the smallest entity that exhibits all behaviors and all parameters of what we know as living systems. Now, this is an interesting thing. Since we assume it is the smallest quantity, we start thinking very small. So, if we look back at the PowerPoint slide now, we start thinking a cell must be this small only. Well, my friends, I am sure some of you do know this and some of you might be surprised to know this, that you are seeing single cells, units of life in your general life also. Think about it. An egg is a unit of life, it is a single cell. Large part of the egg, the yolk and the water, they are actually nutrients for the single cell which is being fed by that particular system, so that it can multiply and grow into an embryo and an organism. So, this way an ostrich egg is a single cell. Many types of mushrooms which grow in the rain around you, many types of these mushrooms are single cells. So, we just discussed a centimeter long cell. Well, believe it or not, you yourselves have a meter long single cell. It goes from here, the top of your head, to the spine. Your neurons are single cells. So, do not think of a single cell as a minimal entity. Think of a single cell as a unit of life. That means, it is the smallest unit or the smallest entity that can be called as a living system. If you break that entity down further, that will not exhibit parameters that form a living system. And what I am asking you to think about is that when you are trying to define, do this exercise yourself, write down what you think are parameters that define a living system, and then try to argue with yourself, and we can have subsequent discussions on this also. Try to argue with yourself in terms of whether, let us say, this object qualifies to be in that parameter or not. So, what you consider as an important feature of living systems may be exhibited by a non-living system also. So, then what is it, which combination of parameters is giving you a living system is something which you have to think about. Learn your lesson from your respected teachers on the IIT PAL system about a cell and a unit of life from this perspective, not from the perspective of trying to memorize names of cells, names of organelles, names of components. Do not do that. Try to understand. The moment you try to understand conceptually, you know, this is the parameter which is going to contribute to making the cell a unit of life. Let us say this parameter is exchange of material from surroundings, then you will get to know what a cell membrane is. You do not have to memorize it, it comes logically. You have to create energy out of the system using a redox reaction. You do not have to remember the name mitochondria and go into a memorization mode. Conceptually, you need a source of energy. Well, and by the way, you have several types of cells which are called prokaryotes, which do not have organelles also. So, you do not have to understand the chemical basis, the molecular basis of living systems by trying to memorize them, understand them in terms of defining parameters, and each parameter will be served by what defines a unit of life. Once you start thinking this way, you will not have to memorize a single aspect of cell biology. You will actually learn how to proceed logically in it. Now, once we have defined a unit of life in this course, the most challenging part found by non-biologists in understanding or studying biology is what I have written here as taxonomy. People see big names, students see big names, even teachers see big names, or even my dear colleagues from other branches of engineering and science see these big names: Escherichia coli, Saccharomyces cerevisiae, Caenorhabditis elegans. The moment they hear these kind of words coming out of my mouth, they get confused and they are like, all biology is about memorization, we can't remember these terms. Well, naming of living forms is called taxonomy. It's very important to understand the basis of this nomenclature. As I shared with you in my introduction to IIT PAL biology, we could see objects as small as 1 angstrom only after 1940s. But then also we were not able to observe biological samples. We can go up to 3 angstrom. We can see small samples now, but we are not able to actually understand the molecular basis of a lot of these things. So, but we were seeing life forms for the last 2000 years, and let us say we see a monkey which is walking like this. Sometimes it is walking on 2 feet, but most of the time it is actually walking on 4 feet, and then we see something like us which is always walking on 2 feet, with 2 hands it is doing other things. Now, we do see that these 2 life forms are producing, they are moving, they are eating food, they are making dirt, they are also living in societies. So, we had to understand the dictionary of nature, we had to formulate a language in which we could put their individual groups inside. You know, we do not go and call this monkey A, that monkey B, that monkey C. We do not go and call this human X, this human Y, this human Z. What we say is, all right, you have 5 fingers, 2 hands with 5 fingers, you walk on 2 feet, you have 2 ears, 1 nose, we will call you a human. Now, this human can have any complexion, but we are all humans. This is how nomenclature started. Nomenclature started from our understanding of what we could see. You will not have to pressurize yourself with remembering names if you follow this concept of where nomenclature and taxonomy started from. Taxonomy started by our observation of features like 5 fingers, things like that. Now, obviously, when we do not have a microscope, we do not see small organisms, so we do not name them, but once we start seeing them, we start seeing some of their activities also. Some organisms move under a microscope, they swim like a fish, some are stagnant, some look round, some look straight, some look cylindrical, some look spiral. We basically started seeing all kinds of forms of these after advent of a microscope, under the microscope. While we were seeing lots of these living systems in a large variety also, we could see all different species of what we called as monkeys earlier. There are monkeys, baboons, gorillas, chimpanzees, orangutans, and we had humans of course also. based on what we saw, what we perceived as common features, we started classifying them in names. We saw plants also, and therefore, we started calling them kingdoms, that anybody who looks like any object which living object which looks like this, we will call it this, we will call it a plant. Any object looks like this, we call it an animal. Within animals, we said anybody who is producing a baby is called a mammal, and so on and so forth. So, we started basically from observation of features. Now, our chemistry has been very strong for the last 400-500 years, but our biology is very young. We have started learning molecular basis of biology only after 1940s and 50s. 1940s and 50s is very young compared to, let us say, 2000 years ago Aristotle, or 500 years ago, where all chemists were really remarkable. From observation of features, as we started looking at molecular components of life, we started looking at observations of functions. What I mean by functions is, I can have this hand grab this object, that is a function. Now, this is easy for you to understand. However, the fact that you cannot digest cellulose which is in grass, but a cow can digest cellulose, that is also a function. We can eat glucose, we can break it into energy, but if we eat grass, the sugar source in that we cannot digest. Cow has other organisms in it, or some features in it. For now, you can basically assume like that some features are present in the cow that allow the function of digesting cellulose, which we do not have. So, it is an observable function now, which we are classifying in terms of digestion of a sugar, whether we can digest one sugar versus the other. So, one part of taxonomy came in terms of classifying that when we digest, when we have a particular function performed this way or that way. So, taxonomy basically is classification, and this will be covered extensively in your chapter, but I want you to go with this logic. It is a classification system based on, it is based on features that provide functions. Today, these features are in terms of alphabetical sequences, what you will learn as DNA or your genome. RNA is also there, of course, DNA, RNA, these are genomes. So, some living organisms have, or some organisms have RNA, some organisms have both DNA, RNA, and most organisms have DNA. The important aspect is that today we classify whether one belongs to a particular group of living organisms or not, based on features that provide functions. These features can be something as an observation in terms of digestion of a sugar, and now we know that this particular sugar can be digested if you have this particular signal coded for digesting it to produce a protein. So, that is DNA sequence. So, essentially we go into sequence which leads to a feature that provides a function, and that is what the basis of our classification systems are. So, taxonomy then it becomes relatively easier for you to understand conceptually rather than memorizing names. So, you now classify, if you start in your mind the concept of I want to classify this organism based on a functional or a functional observation of some function this organism is doing or some function this organism is having, given any kind of features by it, then you start learning with respect to that. You will find taxonomy to be a very interesting field. The reason is, by the way, these features which provide functions, they basically are called phenotypes. You will find this very interesting is, because taxonomy is almost like the language dictionary of biology, in which you have this beautiful diversity of life forms, but you do not get confused. You do not have to memorize the whole dictionary, but you get the basic idea of how to find when a particular feature is seen, whether this organism belongs to this class or that class. I will give a very simple example, a very nice example from our country about this. Many years ago, about 2 decades ago, a team of scientists from India discovered a bacterial species called, it is now called Mycobacterium indica prani. This species is a sister organism, an ancestor of what is today called known as Mycobacterium tuberculosis. But the difference is that this species Mycobacterium indica prani does not cause tuberculosis infection disease, it does not cause the disease, whereas Mycobacterium tuberculosis causes the disease. Understanding that phenotypes of Mycobacterium indica prani, that means how the cell looks, some features on the surface of the cell and inside the cell that provide specific functions are the same as Mycobacterium tuberculosis, but they do not provide disease. Now that has allowed major advances in terms of scientists being able to work on Mycobacterium indica prani without getting infected with tuberculosis and still working on development of kits for diagnosing tuberculosis and helping understand how we can combat Mycobacterium tuberculosis infection. So think about taxonomy, which is found as the most challenging chapter by most students in terms of functional features and phenotypes, and you will find that it is very interesting, not boring and memorization at all. And by the way, you are the generation where you have ample accessibility to all kind of information. Now in that regard, you know, as you go further into biology, nobody will ask you or test you on whether you remember a particular name or not. What will be important is, if you are given a feature, function or a phenotype, or for that matter a name, can you actually logically think where to look for it and how to search these things. This is a very classical way of engineering education. You know, as engineers we were never trained to remember the log tables, we were never trained to remember thermodynamic charts of what will be the pressure at which water will do this versus that. There were always reference charts and tables for us. So what is important for you to learn through your biology textbook is not the name, but how did and why that name came about for that particular organism. The final part of my introductory lecture to you is the only axiom in biology that we know of right now. When I say axiom, it is like a mathematical theorem or a set of mathematical theorems. These are facts and we do not know of any exception to these facts, and they are at the basis of everything that we discuss. The central dogma. If you logically appreciate the central dogma, you will automatically start appreciating the molecular basis of life and you will start getting interested in the miracle of life through logic, not through memorizations. The central dogma was coined by Francis Crick. He was one of the discoverers of structure of DNA. As one of the introductory Know Your Teacher series, you must have seen the pictures of Maurice Wilkin, James Watson, Francis Crick and Rosalind Franklin. Unfortunately, Franklin died. However, it was her data based on which Watson and Crick were able to discover the structure of DNA, and Wilkins had the same data, similar data, and therefore the three of them were awarded the Nobel Prize. Francis Crick was the physicist who was involved in discovery of structure of DNA. Francis Crick was responsible, of course, with discussions with ample number of discussions with great scientists like Watson himself, Wilkins himself, and then Sidney Brenner, who later on went on to win the Nobel Prize for his work on worms. he coined the central dogma in terms of this is the molecular secret of all life. The law of mass action manifests itself into this overall picture. You have DNA, DNA is the secret code. This DNA is converted into RNA. This is like a translation machine, a stencil which will read, help a cell read what is coded in the DNA. This RNA, this transcription or this code reader, after the code has been read, is now converted chemically through a process. You will learn these things in your subsequent specific chapters, into a protein. This protein leads to a function. Essentially, what do I mean by protein leads to a function? Well, your hair is black, that is because some protein is doing this. Glucose was converted to energy, as I mentioned earlier, that is because some protein was breaking bonds of glucose. Glucose is converting to glycogen, that means some protein or set of proteins are binding glucose molecules together. So, protein is, by the way, the phenotype. In a certain way, proteins represent what is going to be the phenotype of a system. The central dogma, as we know it, basically says that all life forms, we do not know of any exceptions, even life forms which have RNA, when they multiply they first form complementary DNA, then again message RNA and then again protein. We have DNA going to RNA going to protein. This series of reactions is called the central dogma. This is the only what we term as dogma, that means this is the only belief, because so far on life as we know it on this planet, we do not know of a single exception to this particular rule of producing life forms. So, essentially when you look at genetics, do not try to memorize terms, think about the logic of central dogma, that you know living systems are coded through a series of reactions. What is that series of reactions? Well, DNA to RNA to protein, that is it. Now, with this concept built into your mind, then you try to understand how things happen, and you will find it is simply fascinating and very easy conceptually to understand. You will learn, for example, that when DNA needs to be converted to RNA, of course, you will have a message first. So, you will have an RNA species called messenger RNA, and then when this message is to be translated into a protein, so you first are transcribing DNA into RNA, then this RNA has to be translated into a protein. This translation is done with the help of what you have as tRNA. So, logically when you go, you will actually realize you do not need to memorize things. Think of it in terms of a series of steps which are involved in the central dogma. DNA goes to RNA, goes to protein, protein performs the function which gives a particular feature or a phenotype to a living system. With this, my friends, I finished my first lecture for you. I hope you enjoyed it, and I wish you all the best in terms of now taking a look at all your subsequent lectures. In each one of them, instead of giving an overall view the way I did now, you will be actually looking at a specific chapter of a specific unit in one of your textbooks, 11th and 12th. Read it, learn from us, and then we will have further discussions. Hopefully, you will start enjoying biology much more than other subjects subsequent to this. Thank you very much.